Our next talk will be by Nick Lane of University College London.
Thank you. Can you all hear me okay? So this title is about as close to philosophy as I would normally get. Turtles all the way down, with this feeling of infinite regress. The last few years in my lab, we've been thinking and doing experiments. I won't show many experiments today, but on the origin of life and using life itself as a guide to the origin of life. And so I'll give you some indications into how this thinking goes. But it's really grounded in what many of you will have heard of as metabolism first, effectively, as opposed to genes first. So before I really get going, I'd like to give a sense of what the shape of the tree of life looks like these days. And someone was saying earlier on sequence and sequence and sequence and sequence. Well, this is what biology looks like these days. There's a tremendous amount of sequencing has gone on. And it looks as if we have the answers to everything. It looks really as if every single millimeter of ecological space has been explored and filled. That niche has been filled with some kind of an organism. And this is a fairly iconic tree of life showing you are here. And if you take a step back from that and you look at the whole tree, well, it's a pretty spectacular thing, but I've written next to it, this is not the tree of life. You could say it's a tree of life because it's a tree of a single gene. And if you were to do the same tree from a different gene, you would get a different tree. It would look a lot like that at a superficial level, but it'd be a different tree. But as soon as you think critically about it, we have the animals up here, the plants across there, the fungi down here, the protists, things like amoeba and so on down at the bottom. And you probably can't read it, but it says bacteria and archaea down there at the bottom in this tiny little space. So that's already telling you that there's something seriously wrong with this tree of life, because the bacteria and the archaea account for a great deal of the genetic variation in the tree. So here we're already looking at the things that we're most interested in, largely the plants and animals. Now this gene, it's a small subunit ribosomal RNA, was the same gene that Carl Woese used about 30, 40 years ago, so around about 1990, with this first depiction of what became known as a three domains tree of life. And this has been described as a Copernican revolution in biology because the eukaryotes, which includes us, so plants and animals and fungi and so on, up here, and then a whole new domain introduced, the archaea, and then the bacteria. So the archaea look a lot like bacteria, and in general, they're pretty simple looking. But in terms of their genetics, they're about as different to bacteria or to eukaryotes as these groups are all different to each other. And there's something implicit in this tree, which took me quite a long time to realize. I imagine a philosopher would see it immediately. But the branch lengths here give you an indication of the genetic variation in these groups. The amount of sequence space that they have explored. So what you can see is that the bacteria and the archaea have effectively explored a tremendous amount of sequence space, rather more than the eukaryotes did. But they remain in their morphology, they're small and they're simple. In their biochemistry and their genetics, they're anything but small and simple. But in terms of their morphology, they are really under an electron micrograph. There's not a lot to see there. So something was happening down this branch of the tree of life, and you can immediately tell that it wasn't linked to information because it's not about searching genetic sequence space. So here's my tree of life. I like to get my head around this one. We have bacteria, which we can see fossils of bacteria or things that look a lot like bacteria from around about 4 billion years ago. And the reason I say they look a lot like bacteria is, well, it's precisely that. They haven't changed. They've been kind of flatlining for 4 billion years. They'd invented pretty much all the metabolism on Earth by 3 billion years ago, including photosynthesis, one of the last things to be invented by the cyanobacteria around 2.5 billion years ago, perhaps. But they really didn't change. And sometime around 2 billion years ago, on a single occasion, it may not have been a freak accident, that's another matter, but it's all the evidence we have says on a single occasion the eukaryotic cell arose. All eukaryotes share a common ancestor, and by definition, we're all related, so all plants and animals and all the rest of it. This is what John Maynard Smith, who was one of the great evolutionary biologists of the 20th century, described as an evolutionary scandal. So the scandal is really all complex life is composed of this one cell type, the eukaryotic cell. Eukaryotes arose just once in 4 billion years, and all eukaryotes share, a textbook's worth of traits. really you get a textbook of cell biology, and you can look at the structure of the nucleus, the structure of the cytoplasm, the endomembrane systems, the cytoskeleton, the lysosomes, all the rest of it. All eukaryotes share them. Plant cells have them, animal cells have them, and so on. And bacteria essentially don't evolve. Bacteria and archaea don't evolve any of those traits. Eukaryotes have them all. So the scandal is that if these traits arose by natural selection step by step, and each step offers an advantage, then why did none of them arise in bacteria or archaea? Well, the essence of the answer is in this tree. This is more of a conceptual tree that goes back to Bill Martin in 1998, and it's rather beautiful. A couple of things to notice here. The eukaryotes are arising from a fusion of these branches, from what's called an endosymbiosis between a host cell that was an archaeon and a bacterial endosymbiont, and that went on to give rise to eukaryotes. We have a second one here, which is the acquisition of chloroplasts from the cyanobacteria, which gave rise to algae and plants. And then going all the way back down, Luca, the last universal common ancestor of all of life, we have the bacteria and the archaea emerging separately from what looks suspiciously like a hydrothermal vent down at the bottom there. So the common ancestor of all of life lived kind of rock-bound inside a hydrothermal system. So as I say, this is from 1998, not so far off, 30 years ago now. And I'm showing it in part because it's become more true rather than less true over that time. I can't say it's completely true, but it was a pretty radical conception when it was first put out, and the evidence that we have now is closer to that than any of the alternatives that were around at that time. So why would an endosymbiosis make such a difference? It's effectively because it changed the structure of cells, or at least that's my take on it. So cells contain mitochondria. The mitochondria are the remnants of this endosymbiosis, the bacterial endosymbionts. So mitochondria derive from bacteria, and they have an electrical charge across the membrane, which is around about 150 to 200 millivolts. But the membrane is only 5 millionths of a meter thick, so if you were to shrink yourself down to the size of a molecule and stand next to that membrane, the field strength that you would experience is about 30 million volts per meter. So that's equivalent to a bolt of lightning. These are the mitochondrial inner membrane, the Christi membranes. This is where respiration is taking place. This is where we're burning food in oxygen to generate all the energy that we need to live. And it's, everyone will have heard of ATP as the universal energy currency, but it's really this charge across the membrane which is driving all the work. It was put forward in 1961 initially by Peter Mitchell. Mitchell had been thinking about these things for at least 10 years before that, and he's here with Jennifer Moyle, who was a brilliant experimentalist and didn't get nearly the credit that she deserves. In 1947 in Cambridge, in this particular picture. In 1957, Mitchell was at a conference in Moscow on the origin of life, along with people like Oparin and Haldane. And here's a lovely quote from him thinking about the origin of life. He said, "I cannot consider the organism without its environment. From a formal point of view, the two may be regarded as equivalent phases between which dynamic contact is maintained by the membranes that separate and link them." So when he's talking about an organism, he's thinking about bacterial cells. And the membrane is the membrane that separates the inside of the cell from the external environment. But it's kind of rather unusual way of thinking about it, that the environment and the bacterial cell are equivalent phases. But if we think about the origin of life, then having phases and having different chemistry across these phases is actually a very constructive way of thinking about the question. So here is what we're talking about. You could think of this as a mitochondrion in your own body, or you could think of this as a bacterial cell. We're taking food, let's just say glucose, breaking it down into what I'm just going to call the Krebs cycle, the tricarboxylic acid cycle, which is basically pulling out CO2 and hydrogen atoms. Not in the form of hydrogen atoms, but let's just keep it simple. It's hydrogen atoms. Those are being split into the electrons and the protons. So we have a current of electrons in the membrane flowing to oxygen. That current of electrons powers the extrusion of protons across the membrane, and that's what's giving rise to this charge of 30 million volts per meter across the membrane. And that is powering work such as ATP synthesis, but all kinds of other things as well. So that's what's keeping us alive now. If you put a plastic bag on your head, the thing that's going to kill you is that process stopping. But it can go the other way as well. So this is not exactly the same figure. This is showing what's called the reverse Krebs cycle. So the arrows are pointing the opposite direction. And the reason I stressed hydrogen before is you can literally pull in hydrogen gas from the environment as H2 and use this membrane potential, use the power of the membrane potential to react it with CO2 to generate Krebs cycle intermediates. And this is a slightly longer cycle than the oxidative Krebs cycle that we're familiar with. It includes things like pyruvate and phosphoenol pyruvate, just for people who love the Krebs cycle. Those are not part of the normal Krebs cycle. But this is the core of all of metabolism. We make, everything makes amino acids, sugars, lipids, meaning fatty acids and things, and ultimately nucleotides from these Krebs cycle intermediates. So growth. What this is doing is turning gases from the environment into stuff that is the stuff of living cells. I'm showing methanogenesis here, but don't worry about that. So I said the bacteria and the archaea are very different in terms of their genetics and their biochemistry. When I say biochemistry, what I actually mean is their protein structures and the way their enzymes are working. So this is the core of metabolism. You probably can't see very much of it. All I really want you to notice is we're starting with CO2 at the top here, and then we've got CO2 in red throughout this. We've also got hydrogen in blue. Really, this is the chemistry of hydrogen and CO2. The whole core of metabolism is about the reactions between these molecules. There's also ammonia in there to make amino acids and so on, and to make nucleotides, but it's really the chemistry of CO2 and hydrogen. And this is conserved in bacteria and archaea. This network of chemical reactions is basically the same in all cells. The genes that are supposed to code for the enzymes that are supposed to catalyze it, they're different, often just not conserved at all in bacteria and archaea, but the chemistry that they're supposed to catalyze is conserved. It seems to be older than the genes themselves. And there's also something which suggests the primacy of metabolism, which is patterns in the genetic code. So in the code itself, this is again starting with CO2 at the top, and this is now just looking at amino acids as these colored lozenge shaped. So in gray, these are amino acids that have got a G at the first position of the codon in the genetic code. And if it's a yellow, then it's got an A at the first position of the codon. If it's green, it's a U, if I remember rightly, and if it's blue, it's a C. I can't actually read my own writing there, but I'm pretty sure that's what it says. So you can see immediately that there's some structuring here of the genetic code by metabolism. And it's really to do with this is the distance from CO2 fixation. The number of steps from the initial fixation of CO2 seems to say something about a patterning of the first letter of the genetic code. If we then look at the second letter of the code, specifically the anticodon, and we organize them by a G or an A, so the closest to CO2 fixation at the top, or a C or a U down at the bottom, this is the hydrophobicity of the amino acid against the base that codes for it. And again, I'm not going to go into the details, but basically it's showing a very straight relationship. The more hydrophobic base encodes the more hydrophobic amino acid, the more hydrophilic base encodes the more hydrophilic amino acid, especially if it's close to CO2 fixation. What that seems to say is that there are direct physical interactions between these things. I won't go into more detail than that, but it looks as if there's something to do with metabolism and direct physical interactions. Down here is a reverse codon table for amino acids, showing it inverted. This is for the third position, and effectively, again, I won't go into details here, but it's saying the third position, when it's not redundant, is about the size of the amino acid. If you've got a small base, you have a bigger amino acid. If you've got the larger base, you have the smaller amino acid. And these are patterns that just shine through the genetic code. So it says there's direct interactions. In fact, what it seems to say is that at the origin of life, the proton motive force, we're going right the way back to the beginning, the proton motive force is driving metabolism, which is giving rise to genes. And if any of that's right, then it says it's all happening in one place, which if you think about a PhD student on the benchtop, that's a very good thing. But it's whether he or she can do it in 3 years is another matter. It's hard to believe that really the origin of life happened in just one place, one hydrothermal system. We kind of want the planet to do the job for us, so it's an uncomfortable prediction. But the kind of environment that might do it were discovered by Deb Kelly 25 years ago in the year 2000, and they're known as alkaline hydrothermal vents. This is the Lost City hydrothermal vent field. These are not black smokers, which most people are familiar with. They're products of rock-water interactions, so relatively cool interactions, called serpentinization. And they are, well, they're not smoking at all. I suppose you could call them white non-smokers, but they are really quite porous as rock. So this is a lump of a black smoker she's holding in her hand, and this is a piece of lost city, and it looks like a mineralized sponge. The hydrothermal fluids kind of wend their way through that sponge. These vents had been predicted to exist actually more than 10 years before they were discovered by Mike Russell. And really, throughout the 1990s, he'd been talking about these vent systems, and nobody was listening. You can look back at the conference proceedings from that time, and Mike's voice would always be there saying, "You've got the wrong type of vent system. You've got to think about these," and everybody would ignore him until the discovery of Lost City. And this illustration here was from a feature article in Nature in 2009, where they photoshopped him up as Erasmus, the Renaissance man, and called him Naissance Man, Naissance as in the birth of life. And there's a little bit of Lost City behind him there. This is the reactor that he built at JPL, Jet Propulsion Laboratory. What he'd basically been talking about, these particular systems are rich in hydrogen gas. You've effectively got hydrogen bubbling out of the ground in alkaline fluids. We have acidic early oceans, rich in CO2. So these two gases that I've been talking about, hydrogen and CO2, are present in this system. And there's proton gradients across these catalytic walls. Proton gradients, the oceans are acidic, the hydrothermal fluids are alkaline, and we've got mixing, but the mixing is frustrated by all of these barriers, cell-like barriers within these vents. And this is a section of Lost City. So we have a very similar topology to modern cells. We have, let's say this is a modern cell, a bacterial cell is pumping protons out. It's more acidic outside. It's got a relatively positive charge outside. And the difference in terms of pH units is equivalent to about 3 pH units. In an alkaline hydrothermal system, we have a much thicker barrier, but nonetheless, we could contrive it as a cell-like space that it's surrounding. Relatively alkaline inside, relatively acidic outside, and with a difference of around about 3 pH units, arguably across that barrier. It's also worth saying it's not just the pores in these systems, it's also the structure of the planet itself. This is a little bit of a forced analogy perhaps, but I rather like it anyway. So the planet, all the iron, which is electron rich, has gone down into the core and the mantle of the planet, so it's relatively negative with all the electrons down there. The oceans and the atmosphere is relatively positively charged, lots of CO2 and the loss of hydrogen and things into space. And the hydrothermal systems are really almost like the pumps in the membranes. This is the connection between the negative interior and the relatively positive exterior of the planet. This is a very cell-like structure. It's almost as if cells are little Earth-like batteries that are recapitulating the overall kind of redox state of the Earth itself. I'm not going to talk about any of our data. This is a time to kind of try and keep it at a high level. This is the hypothesis that we're testing in the lab. There's, I would say, at least some truth to it, but there's also some experiments that are very hard to do. But you can see here we have a pore. We've got protons coming in. It's driving the reaction between hydrogen and CO2 in a reverse Krebs cycle. It doesn't have to be a complete cycle to form lipids, meaning fatty acids, and amino acids. The fatty acids can form a membrane, and we've shown that these fatty acids that can be formed under those conditions will form a bilayer membrane, which will stick to the walls of these hydrothermal systems. So then perhaps we can have biochemistry being pushed a little bit further through to make sugars and ultimately nucleotides, giving rise to what we could call an RNA world, so the beginnings of genetics inside what amount to autotrophically growing protocells. Just one point to make about this really is that if this is true, and I admit that it's a pretty big if, but effectively what this is saying is that metabolism is deterministic chemistry that's going to happen. It's thermodynamically favored chemistry that's going to happen under the right set of conditions, starting with CO2 and hydrogen. And the patterns in the genetic code, and again, we've done some work doing molecular dynamics simulations and NMR work showing that they are real. We can actually measure that amino acids will tend to bind to the bases that code for them in the genetic code. What that would mean is if you have a random string of RNA, then it would tend to template a non-random set of amino acids, which is to say the amino acids that would bind to that string of RNA would depend on the properties of the RNA. Therefore, you would template a non-random peptide to that random RNA. And this is a model done by Raquel, who was a PhD student in my lab at the time. CO2 fixation going through to make these building blocks of nucleotides and amino acids, can polymerize them to form effectively RNA. That can be copied and it can be translated, and translated in inverted commas, but that basically means it acts as a template to make a small peptide, so it's templating that peptide. And these peptides can have functions, for example, in CO2 fixation or as an RNA polymerase, as very, very basic peptides doing that kind of job. And the question is, can you start out introducing completely random RNA into this model and kind of crank the handle around and come up with something that amounts to an optimized code? And the answer is yes. Again, I'm not going to go into this, but this is showing growth rates of these protocells. If you get the conditions right, they will take off and grow. And this is really just the proportion. If it's random, then you'll end up filling the middle space here. If it's not random, you end up with a non-random distribution of purines and pyrimidines, and you can get, under quite a wide range of circumstances, you can get to effectively genetic information with optimized codes, starting with random information. So introducing information into biology, given the reality of physical chemistry, is actually surprisingly easy. And this kind of thing could be very common as well. There may be as many as 40 billion wet, rocky planets and moons in the Milky Way alone. And any wet, rocky planet or moon, assuming olivine is one of the most common minerals in the universe, that's what is doing this process called serpentinization, producing hydrogen gas. We should get similar systems really throughout the universe, and even in our own solar system, these processes seem to be happening on Enceladus, for example, one of the moons of Saturn and Europa, and potentially on Mars as well. So these alkaline vents will have natural proton gradients across cell-like pores. The proton gradients can drive this reaction between hydrogen and CO2. Direct physical interactions will give rise to a genetic code. It's a bit of an alarming prediction to me, but what it predicts is if we find life somewhere else, then not only will it have a similar biochemistry, but that even the genetic code will be related. It won't be the same, but it would potentially have similarities. So life elsewhere would potentially have the same kind of constraints, which brings us then back to this question: why did eukaryotes only arise once in the 4 billion year history of life on Earth? And the answer seems to be this I mentioned already, that it's this endosymbiosis. So the last 10 years or so, we've got much better insights into what the host cell was. There's a whole new group of archaea have been discovered called the Asgard archaea. This derives from the first ones discovered were called the Loki archaea, and they were discovered at Loki's castle, which is a hydrothermal system in the North Atlantic, somewhere between Norway and Greenland. And this was the high point, I guess, of the Marvel universe, and so we ended up with a whole load of archaea. So we have the Odin Archaeota and the Heimdall Archaeota and all the rest of it, and now the Asgard superphylum. But they've now been cultured. They're quite interesting cells with all kinds of long processes and tentacles and things. But inside, there's not a lot going on. They have an interesting actin cytoskeleton. They're definitely complex archaea, but they don't have a nucleus. They don't have complex endomembrane systems and so on, or much compartmentalization. So this is going back to the times when they were first discovered, but effectively what we're seeing then is a host cell, an Asgard host cell with not a lot going on inside, acquiring an endosymbiont, giving rise to the eukaryotes down the line, a cell within a cell. What use is that? This is the one example that we know of cells living within a bacterial cell, a free-living bacterial cell. This is a cyanobacterial cell with thylakoid membranes here. What's the advantage of that? Well, it seems, if we use life as a guide, these are the mitochondria. They go on to become the mitochondria. Mitochondria always retain genes of their own, just a few. Many of you will have heard of mitochondrial Eve. You can trace back human origins to Africa 180,000 years ago or so by comparing mitochondrial DNA sequences. The mitochondria seem to retain only the genes that are required to control respiration locally. This charge of 30 million volts per meter it's pretty serious. You better have a control unit next to it. The mitochondria have got these control units. What it allows them to do is effectively scale up over orders of magnitude for a relatively low genetic cost. So what we see, there are giant bacteria in the world, there's quite a few around, and they always have what's called extreme polyploidy, tens of thousands of copies of their complete genome, and it's usually right next to the plasma membrane. This is a Pulipiscium, which has got that. This has got as many as 200,000 copies of the complete genome, and the genome is 3 megabases, so quite a standard bacterial-sized genome. This is a eukaryotic cell with the nucleus in blue. And then the mitochondrial genomes are green, but the mitochondria themselves are red in this. So we have what I'd like to call a genomic asymmetry. We've got tiny mitochondrial genomes specialized to do bioenergetics, which is supporting energetically a massive nuclear genome. We have about 4 or 5 times as many protein coding genes compared to bacteria, but each one we express 10 to 50,000 times more, which is to say we make 10 to 50,000 times as many proteins from each gene. And we can put some numbers to that. This is from quite an old paper now. And again, I won't go through the details, but we basically have 3 to 5, eukaryotes have 3 to 5 orders of magnitude more energy per haploid copy of a gene. That's the more power to make proteins from genes than, for example, E. coli or Thiomargarita, which is one of the giant bacteria. As power per gram, E. coli has much more power. Per gene or per cell or per haploid genome, it's exactly the other way around because eukaryotes have expanded so much. So why did complex life arise just once in 4 billion years? We're starting with some kind of an endosymbiosis. We end up with a very simple system. The common ancestor is about as complex as this, and there are no surviving intermediates. Something was, there was really nothing was thriving unless they were part of that sexual line that led to eukaryotes. So just for the last few minutes, I would like to talk about one possible reason why that transition was so difficult, because it requires a redefinition of self. And I couldn't resist this with David Chalmers in the audience. He's probably fed up of having this quote read back to him, but this is from his paper in the early 90s. How can we explain why there is something it is like to entertain a mental image or to experience an emotion? It's widely agreed that experience arises from a physical basis, but we have no good explanation of why and how it so arises. Why should physical processing give rise to a rich inner life at all? It seems objectively unreasonable that it should, and yet it does. Well, I know nothing about the central nervous system, but I do think about bacteria, and here's my perception of what a bacterial cell looks like. There's stuff happening in the outside world, and inside is metabolism, and there's really nothing but metabolism going on inside these cells. And if you think about how does this cell respond to its environment, most people in this room will probably immediately say, oh, it's all automated, it's negative feedback responses and so on. There's no requirement for anything that might amount to a feeling. But there's some interesting thoughts that we can have about how this might work. So I've already shown you this. We have a Krebs cycle plugged into a membrane. First thing to notice is everything is in series. We effectively have metabolism, which is putting electrons into this chain of carriers going to oxygen, generating a membrane potential. Everything's linked in series. That's why cyanide will kill you. It blocks that chain linked in series. So, and that's powering metabolism. So we have an interesting circularity that metabolism generates an electrical membrane potential that powers metabolism. It's not a perpetual motion machine. It's kept alive by hydrogen, oxygen, and so on from the environment. But it's balanced by the membrane potential. The delta psi is what it's called, and it's the electrical membrane potential. Bacteria are incredibly active. I find this hard to get my head around. 1 billion reactions every second is what bacterial metabolism is. 1 billion a second, second after second after second. How the hell do they know what their metabolic state is? We could have, I think I did a back of the envelope calculation, and there would be at least 10,000 Krebs cycles spinning every second in a single bacterial cell. Why would they all be going in the same direction? Why would they be synchronized in any way at all? How would a cell kind of integrate its metabolism into a useful state where it can make a decision about what to do? Well, it's the membrane potential. They have to push against this membrane potential, which pushes back against metabolism, and everything is balanced by the charge on the surface of the membrane. So this is really it's got a lot of information implicit in it, which is telling the bacterium how its metabolism is doing in relation to the environment. If there's a metabolic poison, if it's too hot, whatever it may be, if there's not enough oxygen, if there's too much food or not enough food or the wrong type of food or whatever it may be. All of these different processes could have a different automated feedback control that says, some kind of chemotactic thing that says, yes, go this direction. And the other one says, no, go that direction. This one says, no, go down there, whatever it may be. They've got to make sense of this chaos. The way that they can do it is by effectively a single integrated readout of the metabolic state. And the rate of movement of charge, because we have this constant movement of charge. These two things together, the electrostatic membrane potential and the magnetic or electromagnetic fields generated by the structured movement of charge, can give you a great deal of information on your state. Why would you want to know that? Well, part of the issue is that if you start having an automated response, let's say it's too hot, so you activate a heat shock response, a stress response. You start refolding your proteins so that they're getting back to normal again. You've already changed your previous state. You no longer know what your previous state actually was. How do you know how robust you are and what course of action you should take? It's harder for a bacterial cell, presumably, to remember what its previous state was than to have some immediate idea of its current state. So what the membrane potential is telling you is giving you a kind of readout, moment by moment readout of what your state is right now before you've done anything to modify it. And if there's something you can do which is faster than simply changing your gene expression or modifying your metabolic network by phosphorylating everything or whatever you may want to do, if there's something which is useful, which is faster than that, and you make a decision as to whether or not to do it based on your membrane potential, the one thing which is obvious, which could be faster, is do you rotate your flagellum and paddle away? Do you leave? So I would say the first beginnings of feelings is a kind of a readout of your metabolic state in relation to the environment and the beginnings of agency. Should I stay or should I go? Am I all right or am I bad? It's very strictly about the metabolic wetware of cells dealing with a changeable environment and this billion reactions a second and how you know what state you're in. Of course, then we get to come full circle. We end up incorporating these bacteria into a eukaryotic cell. There's a lot of integration goes on. The self of a cell is redefined at the level of the whole eukaryotic cell, not at the level of the bacteria that make up its parts. They have to also come up with some integrated way of dealing with their changes in their environment. The mitochondria need to talk to each other. They need to talk to the plasma membrane, and everything needs to be integrated. After that, we get to multicellular animals, which again imposes another layer of complexity in the definition of a self and how you know what the state of all the component parts are. And then finally, we end up with a central nervous system and the specialization of different neural nets and so on. So I would say that agency and feelings potentially relate to the wetware of metabolism, and that would mean, if it's true, that AI based in silicon is unlikely to have anything resembling feelings. And with that, I'll stop and say thanks to my lab group.
All right, awesome. Thanks, Sean. And it's a real pleasure to be here and a real honor to go right after Nick Lane, whose work I've followed and admired for many, many years. I'm the commentator for The Origins of Life, and there's no better place to start than recapitulating Nick's amazing exposition of how we can think about life as a planetary process in which alkaline hydrothermal vents represent geochemical focusing centers for important planetary disequilibria that can drive life into emergence. In my mind, in these hydrothermal confines, I imagine a web of messy chemistry emerging that is very quickly then sculpted and pruned by the mineral catalysts in those vent walls. This is one of the first ways in which the environment imparts information on the growing proto-metabolic network. Eventually, some kind of autocatalytic network like the reverse TCA cycle will ensue. The biochemistry of today will emerge. You'll transition on and on through what many call major transitions and evolutions, or a series of complexifications that lead up to us, beings of this very lineage that can do science and natural philosophy and ask questions like, where did we come from and are we alone? So again, those primary disequilibria that are at these hydrothermal vent environments are the redox disequilibrium between the oxidized species coming from the atmosphere, CO2, nitrogen oxides, and then the serpentinization driven reactants, hydrogen and methane from the serpentinization reaction, as well as this natural proton motive force, or pH disequilibrium, between the acidulous ocean water and the extremely alkaline fluids. So we can think of these submarine alkaline hydrothermal vent hypothesis as one family of theories about the origin of life with a CO2 fixing engine as its target for research. The paradigm that we're working in is of a life as a dissipative structure, as one of many complex systems that the universe invents to take advantage of free energy gradients in the environment, maintain its own local complexity while raising the entropy of the rest of the universe. And this family of hypotheses is meant to help us explain the unity of core metabolism and the chemiosmotic gradients that our cells are perpetually using and regenerating to perform their functions. Now, as much as I agree with basically everything that Nick said, as the commentator, it is my duty to tell you that this is not the only family of origin of life theories out there on the market. So there is a completely different constellation of theories that posit that our emergence didn't take place at the bottom of the Hadean Ocean, but rather in subaerial ponds. Here, the target of most of these theories is RNA synthesis, where RNA is one of the genetic polymers used in life today. The paradigm generally is that of the RNA world, which posits that RNA, self-templating and replicating, was essentially the first living thing on the planet, and this is meant to explain how we get to our DNA protein world, where RNA plays a vestigial role in translating between the long-term information storage in DNA to the proteins that it encodes. So just a little more detail about this warm little pond hypothesis. The idea is this, that there exists this primordial soup on the surface of a planet with complex organics generated through ultraviolet radiation and lightning in a chemically reducing early atmosphere, as well as exogenous delivery of complex organics from meteorites. Energized by UV light, nucleotides, the monomers of RNA, form inside of this primordial soup, and once you have those monomers, they can be polymerized into functional RNA oligomers given sufficiently concentrating or dehydrating environments, so wet, dry cycles on the surface of the planet are thought to be important to this story, and then RNA polymers then are thought to just evolve Darwinianly, eventually produce cells and biochemistry. So if this story sounds nothing like what Nick just talked about, that's true, and raises the first main takeaway that I want to give you all in this comment talk, which is that the origin of life field faces a challenge in that there are significant divides regarding the environments, targets, and paradigms of origins of life research. Nick talked about one particular environment, target, and paradigm, but there are many out there. So the picture that different origins researchers have in their mind of what the origin of life actually looks like can vary between researcher to researcher. Another interesting thing about the origins of life community is that many papers take the form of this, what I like to call origin of life Mad Libs. Basically, if you have some raw ingredients that you assume were on the early Earth in a blank, blank, blank environment, where those blanks are generally what you can simulate in your laboratory, supplied with your favorite energy source, then you will produce products, which you will then claim are relevant to the origin of life because they are the building blocks of something that we see in life today. And I want to point out the implicit assumption in the scores of papers that take this framework, which is that something that we see in life today was very relevant for the origins of life some 4 billion years ago. But that's an assumption that maybe one should question. So what is universal to life today may not, in fact, be fundamental to what life is or how it emerged. And let me give you a tongue-in-cheek example to illustrate what I'm talking about. If you looked at all phones today and you thought, what is universal about all phones? You might say, all phones have touch screens and cameras. And from that, deduce that maybe the first phone had a touch screen and a camera, in which case you would be very wrong, because the first phone invented in 1876 looked like that. So in more detail, what I'm trying to talk about is what has been often called the event horizon in the origin of life. Many origin of life studies take a bottom-up approach, which is assume some prebiotic scenario based on experimental work and geochemical data that supposedly leads up to the origin of life, the first living thing on the planet. Other studies take a top-down approach to use inferences about the last universal common ancestor, or LUCA, from phylogenetic analyses, the unity of biochemistry, and metabolic fossils. But there may be a vast gap between the origin of the very first living thing and the last universal common ancestor. We don't know how large or small this is. Perhaps there were multiple geneses of life. Perhaps LUCA was actually an ecosystem of organisms trading genetic material through horizontal gene transfer, in which case perhaps non-Darwinian evolution was the paradigm. Often by Occam's razor, we connect the two. We assume that we can learn something about the origin of life from LUCA or vice versa, but again, we need to be cautious about this assumption. And so one question that plagues me when I think about the origin of life is, what about the origin of life can we actually infer from features of the last universal common ancestor, and which features of LUCA might lead us astray? And a meta question is, how would we know which ones? And I don't have any of those answers. I hope some of you have ideas. So in some of my work recently, I've been trying to abstract away from details about the origins of life to see life as one of many complex evolving systems in the universe, where evolution is something that any system with the requisite component diversity, mechanisms for generating combinatorial exploration, and sufficient selection for function are in place. And sometimes during the course of evolution of an evolving system, it may become necessary to start describing that system using the language of novel components that have emerged, new combinatorial exploration mechanisms, or novel selection pressures that become relevant. And during these leaps in evolution, we can think of this evolving system transitioning from one function landscape to another. So in the case of the origin of life, perhaps the first function landscape that we're interested in is that of molecular assembly, whose axes are defined by the static persistence of organic molecules in that prebiotic setting. Some of those organic molecules might interact with one another to form an autocatalytic network. Here we've now transitioned to a different function landscape whose axes are defined not by the static persistence of any individual molecule in that milieu, but by the dynamic persistence of that autocatalytic network as a whole. And of all the different possible autocatalytic networks that you might imagine, some of which might be able to perform rudimentary information processing, sensing the environment, recording memory or information about the environment, and maybe perhaps even acting upon that. And here the axes of this third function landscape might be defined by their novelty-generating capacity. And so I think we have an opportunity here in the origins of life to augment all of the great work that Nick and his colleagues are doing by thinking about what environmental conditions might support open-ended evolution via selection for functions like autocatalysis, homeostasis, and information processing. Because if you don't have an environment that incentivizes or selects for these additional functions, then all you'll get essentially is an organic network that is extremely good at doing thermodynamic dissipation, which I don't think would qualify fully as life. The final point that I want to make in this talk is that I think there's an opportunity from planetary exploration and astrobiology to offer new ways to test the origins hypotheses. And Nick alluded to some of this in his talk as well. So if we go to Jupiter's moon Europa or Saturn's moon Enceladus, these icy ocean worlds, and we find life, that should raise our credence in the alkaline hydrothermal vent hypothesis as a viable origins hypothesis, because we have, at least in the case of Enceladus, some intriguing data to suggest that there are indeed these alkaline hydrothermal vents active in that subsurface ocean, but clearly these types of worlds would not have had a subaerial, warm little pond under a thick, organic atmosphere full of lightning and UV chemistry. Another way of thinking about this is from the other side of the picture, throwing it in the mirror and showing how the origin of life is actually very important for our search for alien life. Why? Because in astrobiology, we aspire to be good Bayesians and try to calculate the posterior probability of having found life on any given world out there. And in Bayes' equation, of course, we have this very important prior term, which encapsulates the probability or expectations for the origin and persistence of life in that environment prior to collecting data that we then want to assess. And so if one researcher has a very different prior for the existence and origin of life on a certain world, that may actually influence and color the way that they interpret data that is returned from spacecraft missions. Okay, so this is my last slide. I'm just going to leave up my take-home messages. Number one, there is a major challenge in the origins of life community in that there are significant divides regarding the environments, targets, and paradigms of origins of life research. There's a big question that I try to think about but don't have any answers to, which is, what about the origin of life can we actually infer from features of the last universal common ancestor, and what features of LUCA might be leading us astray? I think there's an opportunity to search for environmental conditions that support open-ended evolution via selection for function. Another opportunity that astrobiology and the search for life in the universe grants us in informing us about our origins theories and testing those hypotheses. And then finally, this bonus idea that I think is underappreciated, that understanding the origins of life will also greatly aid us in the search for alien life in the universe. And that's all the time I have. I look forward to the discussion later. Thank you very much.